Our Glory Globe TV. Here's a breaking news. Yes, scientists researching to the challenge of high miscarriage rates have been exploring whether it is possible to determine if an egg cell will develop successfully into an embryo or if there is a marker indicating when it is destined to fail. Rutgers-led research teams have found strong clues in two separate studies using both human and mouse data that will allow them to begin to answer yes to both questions. Reporting in Nature Communications, one team found that mouse egg cells that form an unusual cap-like structure before being fertilized are more likely to be viable, attached to the womb and grow than egg cells without the structures. These are important findings because, as many people say, in vitro fertilization for family building success rates are low, said Karan Sindelar, a professor of the Department of Genetics in the Rutgers School of Arts and Science and senior author of the paper Understanding the Basic Mechanism of What Makes a High Quality Egg and Embryo are essential for improving clinical success rate. In the second study published in the American Journal of Human Genetics, the Rutgers-led team identified a gene that, when mutated, causes an abnormal number of chromosomes in mouse eggs, a leading cause of early miscarriage and in vitro fertilization, that is called the IVF failure. We are seeking to understand the genetic roots of female fertility, said Jinsoon Zing, a professor of the Department of Genetics in SAS and senior author of the paper. In this case, the method we developed for identifying genetic risks can be applied by many researchers for further inquiry. Infertility, defined as a inability to conceive after 1 year or longer of unprotected sex, is a common problem in the United States among females ages to 49 with no previous birth. About 1 in 5 or 19% are unable to get pregnant after 1 year of trying. According to the US Centers for Disease Control and Prevention, also about 1 in 4 or about 26% of women in this group have difficulty getting pregnant or carrying a pregnancy to term, a condition known is called as impaired fecundity, the CDS said. Sindelar, Xiang and their teams want to understand how some women produce highly viable eggs and why the process that produces eggs is so error prone. In Sindelar's study, the team zeroed in on one of the last steps of the egg production process. Sindelar said the team was inspired by work on cancer cells by a colleague, Ahana Scope, a geneticist at the University of Wisconsin, who is an author of the paper. Scope discovered that the region that forms between dividing cells contains essential materials such as RNAs and proteins. Because an embryo relies on these essential materials to develop, Sindelar wondered whether a mechanism with life-protecting proteins could also be produced when an egg cell divides into daughter cells. Unlike other cells types, egg cells that divide into two cells from them unequally, one, the egg receives most of the vital materials such as genetic information and the structures that produce proteins, while the second, known as the polar body, receives little and eventually withers away and dies. Using a microscope that produces high-resolution images of living cells, the Sindelar team found that egg cells also have a region between the dividing cells that is enriched in essential materials. In the analysis, they discovered a new cap-like structure that forms between the cells in egg cells that are successfully fertilized and grow into embryos. The caps form protective barrier that prevents the essential materials from escaping into the adjoining polar body cell. In egg cells where the cap was disrupted, embryos were not viable. Benders report. Glory Globe TV, Delhi.